renewal. And also from the stem cells, the cell's origin, it can be differentiated into many type of different cells depending on the microenvironment. Okay, so how the stem cells from the native formation basically differentiate into different type of cells in our body. Basically, it's depends on the microenvironment. All right, and what are the factors, the factors or the environment that favoring the stem cells to differentiate into certain type of somatic cells. And of course, with the stem cells, um, it has a very, excellent proliferative activity or proliferative response. So that if you are using stem cells, for example, to regenerate, or to repair or to replace certain tissues, so stem cell is a good source of cells because it has a very high proliferation activity, which leads to the regeneration of the target tissue. So basically, the stem cells, as I mentioned just now, because it is the native of the cells. So with its capacity to differentiate into various type of cells, depending on the category of the stem cells, and in the stem cells is categorized into 3 different categories, depending on the from where is the source of the stem cells is produced. The source of stem cell is produced. I would love to show you this one. Okay, so you can imagine that the primary source of stem cells is basically coming originate from as early as when the eggs is fertilized, the sperm and ovum. Okay, when the eggs is fertilized, and after the fertilization between the sperm and ovum, it will basically differentiates into more and more totipotent stem cells. So the earliest type of stem cells produced in our body is categorized as totipotent stem cells. So these totipotent stem cells basically type of stem cells basically can differentiate into all types of somatic cells, and can regenerate one whole human body. Okay, one whole human body. So that's the reason why from the fertilized eggs, it basically regrow into blastocyst. So the blastocyst basically is the source of one new human in making. So this blastocyst, from the totipotent stem cell, contain the blastocyst. So the blastocyst, after it's grow and differentiate, it will containing the second category of stem cells called pluripotent stem cells. So okay, so this all right. So this sorry, so the fertilized eggs basically is the totipotent stem cells. So this basically can produce a new human being, another life, another human being. Okay, it can originate into it can differentiate into all types of tissues, cells. And from the totipotent stem cells, after the egg was fertilized, it grow and differentiate into blastocyst. So this blastocyst consists of the pluripotent stem cells. So these pluripotent stem cells also can differentiate into many types of the somatic cells, many types of somatic cells. So this one can form the embryo, okay, from the embryo. So from this pluripotent stem cell, most of the time, this pluripotent stem cell, we can basically isolates from the blastocyst, from the blastocyst stage of the fertilized egg. So if you say that, oh, if we isolate the blastocyst, the pluripotent cell from the blastocyst, can the blastocyst survive? So basically, this I will explain furthermore in my next slides. There are various methods that how people basically can isolate the pluripotent stem cells. So from the pluripotent stem cells, it will isolated into it can be isolated and cultured, okay, which I will explain later how, and also differentiated into many types of cells, many type of somatic cells, hematopoietic, neural to produce neural stem cells to produce neurons, mesenchymal stem cells to produce bone cells, connective tissue, cartilage, the all sorts of human body, and um, and those type of cells basically are categorized as the multipotent stem cells. So how, sorry, I have to go back to the previous slide. So different category of stem cells basically describes as their capacity to differentiate based on the capacity of their differentiations. So as I mentioned to you, totipotent basically can develop into one new human being, one new individuals or human being. And of course, the pluripotent can form more type of cells, even it's not complete, even it's not complete as individual, while the multipotent basically have limitations in terms of the type of cells they can differentiate it depending on the source of the cells are isolated. So for examples, multipotent, for example, we can isolate the multipotent stem cell from the fetal tissue, meaning that from your bone, we can isolate from the bone marrow, we can isolate the multipotent stem cells from the cord blood. The cord blood, when the baby was born, you have the cord blood, and also from the adult stem cells that can be isolated from the blood, from even from the saliva as well. You can isolate the stem cells. Sorry. Okay, so from this category of stem cells, they are divided into several types. Okay, they are divided into several types. So the first one, the sources of stem cells, the first one is, as I mentioned just now, the embryonic stem cells. Embryonic stem cells are isolated at the totipotent and also pluripotent level, which is during the pregnancy stage, during the pregnancy stage. So you can, as I mentioned just now, from the blastocyst, in the donor program, for example, in USA, United States of America. So in the donor program or in the IVF method, um, IVF method, you know that we can do the in vitro insemination procedure for those having problems to have baby. So when they do the IVF method, sometimes there are blastocysts that are no longer, they are not well graded to be continued for the pregnancy, for implantation. So there are programs, the donor programs, whereby the blastocysts will be that are graded low will be used for the isolation of pluripotent stem cells. So they will donate the blastocysts for the stem cell program. And the second category of stem cells is called adult stem cells. So these adult stem cells are at the category of multipotent stem cells. So you can isolate the adult stem cells at various location in your body, okay, from the bone marrow, from the fatty tissue, for example, via the liposuction procedure, whereby people who overweight and they go for liposuction procedure. So the fatty, the so uh, the fatty as the apa the lipid cells can be used as a source for the multipotent stem cells. All right. And also the next category, the third category of stem cells is called induced pluripotent stem cells. So the induced pluripotent stem cells basically is somatic cells that you can isolate, for example, from your skin, okay, epithelial cells, given certain growth factor, and the skin cells can differentiate into induced pluripotent stem cells. So let's have a look at one by one so that you can understand what are the differences. So embryonic stem cells, as I mentioned just now, derived from human embryos by harvesting the inner mass of donated excess IVF embryos. So basically, when the couple or an individual undergone the IVF procedures, in vitro insemination procedure, so they will basically having several sets of blastocysts. Okay, several sets of blastocysts, whereby they will grade it from good to poor. Okay, based on the blastocyst criteria. The blastocyst is the cells. So, so every blastocyst will form individual cells inside the blastocyst. So those blastocysts with a highly graded category will be selected for the implantation, for the pregnancy, whereby those with poor grade will be either aborted or maybe go to the um, donor program, the stem cell program. So this is what we use in the collection of the embryonic stem cells. And also via the somatic cell nuclear transfer, if you went through the um, okay, so I have the image here. So if you went through the cloning technology methods before during your undergraduates, I think you have learned about the somatic cell nuclear transfer method, whereby you can clone cells in the presence of the donor cells, okay, in the presence of donor cells. So either collection via the first method or the second method, okay. The first method is via IVF, you know that. So this one is a natural process. It's a natural process whereby the natural process was basically is done in the laboratory settings. In the laboratory settings, that does not need any induction or any treatments because it mimics the process happen naturally. Okay, however, it is conducted in a lab. But in the second category, somatic cell nuclear transfer, it needs the donor and also the donor cells and also the egg, whereby um, the egg is basically is a donor, is a donating the genetics material, donating the genetic materials for the donor cell to carry the and replicates and multiply in a somatic cell nuclear transfer method. So with this SCNT method, the the fertilization, the fertilized eggs will be developed into blastocyst. Okay, will develop into blastocyst, and the blastocyst can be used for the collection of embryonic stem cells. And okay, so when we collected these embryonic stem cells, most of the time in the laboratory setting, they are grown in the undifferentiated states, meaning that in the embryonic state. So it retains ability to specialize into all the different tissue types. So if they are able to retain the differentiation capacity into all type of cells, so they are categorized as they are if they are able to retain a category to all type of cells, they are categorized as totipotent. However, if they are ability to differentiate into specialized type of cells, meaning that not all type of cells, they are categorized as pluripotent. Okay, pluripotent. Okay, so why do researchers want to use embryonic stem cells along with other technologies? Okay, so basically, the difference between the pluripotent or the embryonic stem cells, embryonic still in the category of pluripotent stem cells, because their abilities to differentiate into various type of cells, more than what the adult stem cell does. Okay, more than what adult stem cells does. So these adult stem cells, which referred to the multipotent stem cells, they have limitations in terms of the differentiation capacity. So um, in terms of the advantages, of course, the embryonic stem cells has more advantages compared to the multipotent stem cells, because it can proliferate indefinitely in culture, very high proliferation level, and also it's easier to obtain than adult stem cells. It is mentioned easier because if you have the donor program, for example, it is easy to collect the embryonic stem cells because it does not invasive. It is not invasive, and you don't have to hurt the subjects in order to collect the embryonic stem cells. However, in a condition where you don't have any um, donor or the stem cells program, it is not easy as I mentioned just now, because you have to ask people to make donation of their blastocysts, which is it is not easy in certain countries, especially those who has the very strict Islamic regulation. Very strict Islamic regulation. It is it is not possible to have this kind of the donor program. So in the next stem cells category, which is the adult stem cells, or known as the multipotent stem cells, also is called as somatic stem cells, because they are isolated from the somatic cells. It includes the stem cells isolated from fetal tissues, from fetal tissues, and umbilical cord. Okay, this one is another stage, whereby after the baby developed and grow during pregnancy into fetus stage, and after their birth, for examples, I think some of you have heard the cord blood cord blood cord cord blood bank. Sorry, a cord umbilical cord banking, umbilical cord banking, whereby you can store this umbilical cord from your baby. That will be stored as a source of stem cells. So in terms of when you store this umbilical cord blood, so you can isolate the stem cells, which is beneficial for you in the future in the case that you have encountered certain diseases, for example, leukemia or certain other diseases that needs regeneration of your tissues. So basically, you can use this kind of a stem cell source to repair your injured tissue. Okay, so and also these adult stem cells are involved in tissue homeostasis and repair. I just mentioned just now. So they are generally multipotent. So when we call multipotent, they have limitations to how many type of cells they can differentiate it to. Okay, because they cannot differentiate into all type of cells. So they can differentiate into a specific type of cells depending on their lineage. Lineage meaning that from where it is originate, from where it is originate. So difficult to isolate and grow in large numbers in the laboratory. So let's have a look at one by one. So example of the adult stem cell, ASC, the first one and the most easiest to isolate is hematopoietic stem cells. So these hematopoietic stem cells are blood origin, So they are used to produce our blood cells, including the B cell, T cells, and also all the leukocytes, all the leukocytes. So this one is the most easiest to isolate. It can be isolated via your blood or via the lymph nodes, the lymph nodes. And the next one is the mesenchymal stem cells, reported to be present in many tissues. So this mesenchymal stem cells is a type of stem cell that I use in the lab. So these mesenchymal stem cells are largely reported to be isolated from the bone marrow, um, via the bone marrow stromal stem cells or skeletal stem cells. And also this lineage of the bone marrow stem cells basically can differentiate it into these skeletal cells such as bone cells, cartilage cells, which is the chondrocytes, or the fat cells, and also the stromal cells that support the blood formation in the skeletal system. And the next one is the neural stem cells. Okay, some cell source can basically differentiate into neural stem cells. Some cell source can be differentiated into neural stem cells depending on their lineage. Lineage means from where its origin, as I mentioned, mesenchymal stem cells, they can originate from the bone marrow, So from the bone marrow, it can differentiate it into many types of skeletal cells or hard tissues cells, um, including the tooth. And neural stem cells in the brain give rise to its three major cells. So neural stem cells in the neural stem cells, basically their lineage is very limited, so they can differentiate it into all the all the brain cells, which include the neurons and also astrocytes and also oligodendrocytes, which is important in the brain system. So you can have also isolated the stem cell from the skin. This is also quite common. However, quite common, but exhibit quite number of limitations. That's why it is not widely used as a source of stem cells. So as I mentioned just now, you can isolate the cells from the bone marrow. This one is the bone. You have the bone marrow. So from the bone, you can basically isolate the hematopoietic stem cells, and also the multipotent stem cell, or the MSC, mesenchymal stem cells, which can be differentiated into various type of cells, various type of cells. From the bone marrow, you can have the blood, the hematopoietic stem cells, and also the adult stem cells. For example, those that can be differentiated into the skeletal cells, skeletal cells. And the last category, not category, the last type of stem cell is called induced pluripotent stem cells. So these induced pluripotent stem cells, basically, they are not stem cells in origin. They are not stem cells in origin, so they must be induced from the adult cells, from adult cells. For example, from epithelial cells, the skin cells. You isolate your skin cells from your cheek, inside your oral environment. So with the help of certain factors, which the system was developed by Shinya Yamanaka in Kyoto, Japan. So this Shinya Yamanaka from University of Kyoto, he included 4 specific genes as a transcription factors to convert the adult stem cells into induced pluripotent stem cells. Okay, so this researcher, Professor Yamanaka, he is still alive until today, and he received the Nobel Prize award in 2012 together with the Sir John Gurdon for the discovery that mature cells can be reprogrammed to become pluripotent. So this Sir John Gurdon basically is the inventor for the cloning method, the somatic cell nuclear transfer method. Okay, so they share together the prize for the Nobel Prize Award. And with these induced pluripotent stem cells, as I mentioned just now, the origin cells basically is the somatic cells. It's a common cells that we have in our body. You can isolate cells from your urine, from your saliva, from your skin. So these cells, with the help of 4 factors, the CMYC, OT4, KLF4, and also SOX2, in combination can induce the somatic cells to behave like a stem cells, So you must be wondering how we differentiate the somatic cells and the stem cells. How we differentiate the somatic cells and stem cells. So when we have cells in our body, different type of cell, we have many cells. Even though you isolate one tissue, for example, you have bone surgery, for example. We have bone surgeries. So when you isolate cells from the bone environment, it's not only bone cells that you isolate. Okay, there will be blood cells, there will be marrow cells, that will be some muscles, okay, fat. So how we differentiate one cells to another? Okay, so we can differentiate cells one to another by looking at the marker expression at the cell surface. So different type of cells, okay, will have a different markers or the growth factor or the transcription factor expressed by the cells. So the most easier to differentiate, to characterize the cell is by looking at what are the factors or the markers expressed at the surface of the cells. So that's how we differentiate the stem cells with the somatic cells. Because stem cells and somatic cells, they don't share the same marker most of the time. Okay, they don't share the same marker, the same protein on their surface. So these induced pluripotent stem cells, as I mentioned just now, so you can have it from the patient. Okay, you can have the personalized medicine techniques, method, whereby, for example, this patient having cancer, for example, having cancer. So you can isolate the cells from the patient, the skin cells from the patient. All right. So from the patient itself, you can induce into pluripotent stem cells. So you produce patient-specific induced pluripotent stem cells. And with the cells that you produce, it is pluripotent stem cells. You know that pluripotent stem cells can differentiate into various type of cells. You can use that to repair or to, for example, to repair or to promote the production of more and more healthy cells in the patient system, in the patient body. Okay, or you can use the cells as a carrier to carry the treatments to the patient's body. All right. So this induced pluripotent stem cell can be maintained indefinitely in the lab. Okay, you can maintain because it is pluripotent. So it has a very long shelf life, a very long shelf life. And pluripotent with similar properties to embryonic stem cells. So as I mentioned just now, it has the advantage because it is in the pluripotent state. So you can differentiate them into multiple type of cells. However, there are pros and cons. There are pros and cons to iPSC technology. The first one, of course, the technology is expensive because you have to maintain and induce them with the help of the transcription factors. So you have to do the treatments. So meaning that it requires more cost compared to the natural stem cells. The pro is basically cells will be genetically identical to patient or donor of the skin. So as I mentioned just now, so if you have patient and the cells you isolated from the patient directly and do the induction. So basically this one can avoid the immune rejection because you are transferring patient own cells. Okay, patient own cells, so dia tak ada, it will not cause any graft versus disease rejection. Okay, no rejection. And of course do not need to use an embryo because even though it is the pluripotent, but the pluripotent basically was induced from the somatic cells, okay, from the adult cells. And the cons, the contra, basically, when you induce the cells in an in vitro system, so the cells might carry the genetic defects. Okay, so for example, if you isolate the cells from thalassemia patient, okay, so the thalassemia basically is a genetic disease. So the cells still carry the thalassemia trait. The cells still carry the thalassemia traits. And the one of the pluripotency genes is a cancer genes. So from the 4 genes here, CMYC, OT4, KLF4, and SOX2. So the KLF4, if I'm not mistaken, is the cancer genes. It's a cancer gene. It's a gene that can cause cancer. Can cause tumor development. All right. So viruses might insert genes in place we do not want them and causing mutation. So when you do culture in the laboratory system, so you don't know what infected your cells. So it might carry some sort of infection with virus, with fungus. So yeah, there will be some insertional genes from the environment and cause mutation. So after we already identify various type of the stem cells. So how the stem cells is processed in the laboratory. So it involves four processes. It's involved four processes. The first one is isolation. The one is what I described just now. So you can isolate the stem cells from various type of sources, from the blastocyst in pluripotent, from the bone marrow, or from the blood as multipotent stem cells, or from the epithelial cells that you induce into the induced pluripotent stem cells. So when we decide from where the location of for the isolation, so you have to make sure that the cells retain their functionality and potential to differentiate. So when we isolate the cells, okay, so the first thing that we have to do is to confirm there are stem cells. So how to confirm there are stem cells? So we have to do the characterization, okay? So characterize and track stem cell population, okay? Uh, okay, characterize and track stem cell population. So most of the time in the lab, like what we did in our lab, so we will do the surface marker characterization by looking at by looking at the specific stem cell markers. Okay, specific stem cell markers. For example, CD105, CD, kalau hematopoietic stem cells, they should have this CD41, 42. Okay, so this CD is a cluster of differentiation or the protein at the surface of cells, So because this is master level, I don't have to go back to the basics, right? So CD, I think you have learned CD4, CD8, kan? CD4 is exclusive for the T helper cells, while the CD4 and CD3, CD4 is a T helper cells, while the CD8 is a cytotoxic T cells. So different type of the CD or the surface markers or the protein on the surface basically will characterize the cells into different type of cells. So, and also we have to make sure that the cells are transplant ready, meaning that they are still in the shape or in the format of stem cells. And after we characterize them, we have to expand, do the expansion, meaning that make it more and more in numbers, make it more and more in numbers. So culture stem cells in a stable, multi or pluripotent state, free from mutation and to sufficient quantity. So we have to make sure that the system to expand the cells, or expand mean not to make it large, to make it more, to make it higher in number. So we have to make sure that it is free from mutations. So you have to do the genetic screening, as well as to make sure that it's sufficient in quantity before you use the stem cells for the uh, next procedure. Okay, next procedure, and enable economical expansion to make cell therapy a reality. So yeah, there is a challenge to assure that the processes involved in the stem cell procedure is economical. All right, so we have to make sure that we are using a minimal amount of treatments just to maintain the stem cell state. And the next one will be the differentiation. Okay, depending on what type of the tissue that you would like to regenerate or you would like to repair, so you have to control and activate the stem cell differentiation to desired lineages. So when we work on stem cells, we are working on the stem cells in the control environment, in the control environment. So if you want the stem cell, for example, to be differentiated into bone cells, for example, so you have to make sure that you are giving the you are fit the environment similar to the bone micro environment, to bone micro environment. All right. So if you want to differentiate into bone, into neural cells, so the environment is totally different. Environment is totally different because our microenvironment also is different. There are different factors, there are different transcription factors, um, there are different biochemicals needed to promote the differentiation of the cells. For example, in bone, because I use more in towards bone. For the cells to be differentiated into bone, it needs the phosphate. Okay, it needs the phosphate. It needs to have the calcium, so that it can regenerate into the bone-like cells. So, yeah, I give example. Okay, these are few examples. So these are the cartoons. So let's have a look at this one first. So I give example if you want to isolate from human umbilical cord and also fetal placenta. Okay. So anyone married here and gave birth already? Okay, yeah. So this one is the cord. It's an umbilical cord. It's an umbilical cord that attach to baby, that attached to baby. Okay, so when the baby, when the mother gave birth to a baby, so you can collect this umbilical cord as well as the placenta. This one is a placenta. Okay, as a source of your stem cells. So this one is an umbilical cord. So this umbilical cord, you have to um, open it and scrape off the tissue. Scrape off the tissue to isolate the stem cells. Isolate the stem cells. So from these two sources of tissue, you can have the various type of cells. Various type of cells. You can have the cord lining, water jelly, cord placenta junction, and also fetal placenta punya cells. So you can slice out all the tissue from here, and with the help of the enzymatic digestion procedure, for us, we are using, you can use trypsin or you can use collagenase just to break all the tissue and release the cells. Release the cells. This is what we are doing in the lab now. But for now, I am processing the cartilage, and isolate the chondrocytes from the patient cartilage. So we are using collagenase, enzymatic reaction, collagenase, or you can use also trypsin. It's also enzymatic reaction, and the cells will be removed from the tissue, and you can isolate the tissue in a culture flask. In culture flask, I think you will have the research project after this. Okay, that will be clearer for you. And after isolation, you have to do the characterization, as I mentioned just now, to confirm the cells that you isolate is the stem cells. Okay, so you can use morphological evaluation by microscopy. You can use the analysis of markers, as I mentioned just now. It's a surface marker or the surface protein on the cells using flow cytometry. And also you can use the marker as well by real-time PCR, detect the expression of the genes from the cells. And also you can do the multi-lineage differentiation. Okay, so from one type of stem cells, you can test them to differentiate into various lineages, whether they can differentiate into bone, into cartilage, into fatty acids, for example. And then after all this already passed, okay, so you can use them for various potential application, such as for the biotech, pharmaceuticals, cell therapy, and also for the regenerative medicine to regenerate some tissues. And also, as I mentioned just now, you can also isolate from bone marrow, okay, and also from the lipoaspiration, from the liposuction procedure, and also you can isolate from the tooth, from the extracted tooth, okay. So from bone marrow isolation, as I mentioned, similar process it will carry, okay. It's just that from bone marrow aspiration, you don't have to do the enzymatic digestion because you only isolated the cells, but you have to do the separation. So that's why in the bone marrow aspiration, there is no collagenase or enzymatic punya procedure, but you just need to do the separation using Ficol. Kalau you belajar, if you have learned immunology before, so from the peripheral blood mononuclear cells, you can separate them from the RBC, from the red blood cells or erythrocytes by using the Ficol by a constant centrifugation. And you can isolate the PBMC, so your cells will be isolated at the PBMC region, at the PBMC punya phase, okay? And you can do the expansion and of course characterization to have a look at the surface marker. And also lipoaspiration, you can do, okay, from lipoaspiration, this is also quite common. So you can have whenever you want to collect from an individuals or subject or human, so you have to make sure that you have the ethics. You have the ethics, meaning that you have to seek permission from the donor. So the donor has to give you the consent to allow you to collect even their waste, like in lipo suction procedure. So when they aspirated the fatty acid cells, the fatty cells, the adipose cells, sorry, we call it adipose cells, okay, adipose cells. Even though it is a waste from the procedure, still you need to get the patient consent. So from the adipose, you have to do the collagenase digestion, macam similar to this one. So collagenase digestion or trypsin is also one of the preferred method, and you can do expansion and characterization. And from the tooth, okay, from the tooth, there are number of location from your tooth. You can isolate the stem cells. You can isolate the stem cells. So from the tooth, you can isolate from the apical papilla dekat sini. You can from the periodontal ligament dekat atas sikit, up a little bit at the ligament, and also from the alveolar bone, from the bone that attach, because our tooth basically was attached to the bone, attached to the bone. Okay. Interestingly, from the some parts of the stem cells isolated from the tooth can be differentiated into neural stem cells, into neural tissue, because their lineage is basically same with the neural cells. Okay. So you have to do this collagenase and also expansion and also next characterization before it can use for the various applications. Okay, so as I mentioned just now, when we do the characterization, so most of the time we go for the stem cells for the characterization using the stem cell markers, So the most common markers that we use to identify as a mesenchymal stem cells are CD75, CD90, and CD105. So this CD basically is not expressed by the immune cells, by immune cells. It's not expressed by immune cells. It's only expressed by the stem cells. Okay, it is negative to CD14. Okay, CD14 is a macrophage. Yes, macrophage. CD34 is a hematopoietic cells. 45 is a T cells. CD19 is a B cells, and also HLA-DR. So it should be, when you tested your stem cells, it should be tested negative for all this, because all this represented the blood cells, blood cells. All right. So what makes stem cells so valuable? Okay. As I mentioned just now, in a condition whereby we need a lot, we need more, and our body is no longer can produce a sufficient amount of cells to repair, to replace, or to regenerate. Okay, there are certain conditions. For examples, when you I give examples. When you have injuries to your cartilage, cartilage. It's not muscles, it's cartilage. On your knee, there will be the cartilage, which basically cushion. Uh, uh, um, its roles as a cushion, as a cushion to make sure that this is your knee, okay? When your knee move, it must have a cushion between two bones, okay? When you don't have the cushion, then your bones will be, touching each other, and you will feel pain, okay? You will have pain. So when you have injury to your cartilage, for example, on your knee, and this one is in your knee, and cartilage in an environment whereby it has a very lacking of the blood supply, it has lacking of blood supply. So to regenerate the area is very, very difficult, because the microenvironment is not promoting for the regeneration of the cartilage. It's different to bone, okay? Bone, if you have if you see somebody in the ward who have a car accident, and broke their leg, the bone basically has a very high capacity to regenerate, because our normal bone, even though we don't face the car accident, is always replaced, is always renewing, it's always renewing. The bone that we have now is always a new bone, because the cells is always renewing. The bone cells is always renewing the tissue to produce a new bone tissue. We cannot see that, but that's the nature process occur in our body, for bone. So if you have injury to bone, it is much easier to be regenerates or repair compared to the other sides of our body, for example, the cartilage. So in cartilage condition, because cartilage is lacking of the blood supply, and it has only getting the supplies from synovial fluid. So it is almost impossible for the cartilage to be regenerate. And when you have injury to your cartilage, you will be on the painkiller throughout your time. Okay, throughout your time. So one of the alternative that people are opt to now is to go for the stem cell therapy, whereby you inject your knee with the stem cells to help regenerate the environment, regenerate new cartilage to the environment. So that's one of the scenario that we can use the stem cell. So we can use that for the cell therapy, transplantation of specific and precursors. And of course, we can apply the stem cells for research, to look for the new drugs, to look for the possible, or use that as a model to understand the behavior or the the genetic changes of certain cells or system. And as I mentioned just now, you can also use the stem cells for the development of new drugs. Okay, so in stem cell based regenerative medicine, okay, so when we're talking about regenerative medicine, either we use the cells individually, or sometimes we couple the stem cell with the biomaterials, with the component, a carrier, a scaffold, so that the cells can be differentiate and regenerate to form into certain structure, So okay, so mesenchymal stem cells basically is also um, other than the differentiation capacity into various type of cells, it is also secreting some immunomodulatory factors, immunomodulatory factors. Okay, so by communicating with the immune cells, so these mesenchymal stem cells can also having this one is immunomodulatory, the spelling mistake, immunomodulatory effects, whereby it can communicate with the immune cells to helps, to helps protect us the environment, to help protect the environment. And also by secreting some of the trophic factors like cytokines, chemokines, and also exosome. Okay, I'm not sure whether you have separate lecture on exosome, but exosome is actually is a byproduct of cells. It's a byproduct of cells. So exosome can be in forms of proteins, RNA, DNA. So exosome has some roles in the cell-to-cell communication. Cell-to-cell communication. So by secreting the exosome, by secreting the exosome, so these mesenchymal stem cells can help the cells in our body to communicate and give signals to help regenerate or repair the environment or protect the environment, like what it does with the immune cells. And of course, it has the migration and homing capacity, whereby it is important in the repair mechanism, whereby when you have injury to certain area, or like I mentioned just now, when you have injury to your bone cells, you break your bone, so these stem cells can help to give signals for the migration or homing of the surrounding cells to produce and repair the injured tissue, repair the injured tissue. So regeneration in human, so at the moment there are numbers of the reported studies, and also some country already approved the cell therapy using stem cells to be used for their patients. Okay, so we are able to regenerate various types of tissue. So the most commonly used regeneration procedure is for the skin. This one is the most simplest. For example, in a burn patient, ataupun in certain like injury to the skin, like with acid splash. Okay, so you can regenerate the area with this stem cells help. Okay, and of course the more regeneration in human is the colon. It's a colon. It's a colon. So you know that when people suffers with the colon cancer, for example, the colon can be cut, the cancer site can be cut, and the colon can be repaired very fast. Can be repaired and healed very fast. So my auntie experienced colon cancer 10 years ago. Okay, she got it cut and she is well until now, and it is how the colon can regenerate very fast in our body, and of course the our liver, so the liver is also the fast regenerates tissue. Okay, so even you can donate some parts of your liver to somebody, and it can regenerates very fast. And the moderate regeneration is bone. As I mentioned, bone is always regenerate. Why it is in moderate? Because to produce one whole bone, it will take times. Because one whole bone, it is a very structured tissue. It's skeletal tissue. So to have it regenerate and having functional as the previous bone, it will take some time, okay, and also the muscle ligament, and the low regeneration capacity of tissue in our body is the heart. If you have infarction, where your heart tissue died because of infarction, so most of the time there is no turning back. Most of the time there is no turning back. And also, as I mentioned just now, we have the image here, the cartilage. So we have the cartilage, that cushion between two bones. This one is our knee. For example, this is our knee. This is between two bones. Okay, this one between two bones. It can also on your elbow. It can be on your elbow. So if we don't have this cushion, we'll have pain all the time, because the bone and bone, when they having in contact with each other, you will have pain. All right. And of course, the brain. So mesenchymal stem cell research statistic, as you can see here, so the data is not very new, but if you can see that from the 1992 to 2015, similar statistic, so you can see that how it is, you know, um, the research in these mesenchymal stem cells or stem cells increase. If you can notice that it started from the 1990s, whereby people, when the people know molecular biology, okay, when the people have more and more methods and techniques in molecular biology. So we have the first cloning in 1996, the Dolly. So when they have the cloning, so people start to think of what are other source of cells that can be used for the cloning procedure, from the cloning procedure. So yeah, this is the advancement in our research, advancement in the technology, and also to the therapeutics methods to explore the possibilities of mesenchymal stem cells to be used for the treatments. So if you can see the US stem cell market, of course, US is leading. They have very well regulated with FDA and so on. So you can see how big is the business in stem cells. So they have increase in the market or demand for various type of stem cells. Various type of stem cells. And growth in novel application of stem cells. Okay, so if you can see that we have so many clinical trials, even the numbers is increasing every year, and the regulation is also, the country is also having their own regulation for the stem cells clinical trials and also therapy in the country. Disease areas utilizing stem cells for research or drug discovery application. You can see the most is the oncology, the cancer. So the most, if I'm not mistaken, is a blood cells lah. It's a blood cancer. It's a blood CA. The most utilized, and also like I mentioned just now, it's a colon cancer whereby you have the cut and you want to regenerate the area, so you can also apply the stem cells. And diabetes, so diabetes most of the time involve the removal of certain tissue or certain tissue, and the tissue needs to be replaced. So that's why diabetes having a high demand for the stem cells. Neurological disorders like Alzheimer's, like Parkinson's, they are also considering using the stem cells to using the stem cell as a cell therapy to help regenerate new bones and new brain cells, especially in Parkinson. Because we know that Parkinson, the patient are losing their ability, uh, the sensory, and Alzheimer's losing their ability to memorize and remember. So they are opt for the stem cell therapy. Yeah? And bone and skeletal disease, as I mentioned, inflammatory disease, cardiovascular. So if you can see, the demand is higher for the easiest regenerate tissue. So cardiovascular. Oh, Alzheimer's is here. So Alzheimer's has its own study, statistic or data. It's not putting under neurological disorders. And Parkinson's, psychiatric disorders, and also respiratory, pulmonary. And also, as I mentioned, when we talking about the regeneration of tissue, most of the time, it will apply the tissue engineering methods, tissue engineering approach. So in tissue engineering approach, the main aim is to replace or repair the damaged tissue with the help of three factors. With the help of three factors, the cells, you must have the cells, either the somatic cells or the stem cells. The growth factor or the signals can be is come from the growth factor or transcription factors, or drugs you can add in the during the treatments, and also the the extracellular matrix. ECM is extracellular matrix. So this extracellular matrix basically uh, uh, act as a scaffold, as a base, okay, as a base or scaffold for the cells to grow. Cells to grow. So for example, you now regenerate the bone, for example. When you regenerate the bone, you kena ada matrix. You need to have a matrix. You have to have the scaffold, the base. Macam like we bake the cake. You must have the container for the cake. All right. So similar to when you want to regenerate certain tissue, like a very structural tissue like bone. So you need to have the scaffold or the matrix. So the scaffold or matrix can come in the form of metals, ceramics, synthetic polymers, and also natural polymers. So stem cell in regenerative medicine, okay, so to promote the repair response of disease, dysfunctional or injured tissue using stem cells or their derivatives. So in the regenerative medicine, the main aim basically is to promote the repair of disease, dysfunctional or injured tissue. Disease means meaning that if you have tumor, for example, you have bone tumor and you remove certain part of your bone, so the bone has to be replaced so that you can still function as normal, or dysfunctional. So dysfunctional can be because of the from the birth defects or dysfunctional due to genetics that come later in your life. So you can basically try to repair the dysfunctional side of your body and also inject tissue. For example, if you have accident or if you have infection at that site, and the infection causes losing of certain area of your body, so you can regenerate or repair the area with the stem cells. So it is the next chapter in organ transplantation and uses cells instead of donor organ, which are limited in supply. So what is the most advantage of the stem cells? Basically, stem cells, because it can regenerate tissue, it can it can be used to as alternative for the organ donation. Okay, yeah, in certain condition, definitely you need the one whole organ donation. For examples, if you have kidney failure, okay, if you have kidney failure to regrow or regenerate one whole kidney is impossible. It's impossible because in regeneration, the aim is not to regrow or regenerate one whole structure. The portion, okay, the portion. Kalau bone, the portion of your bone. Um, Still, in certain condition, you cannot opt for the regeneration, like in the kidney failure. So in kidney failure, when you have kidney failure, both fail, left and right, you have to find for the donor, or else you have to go for the dialysis the whole throughout your life, throughout your life. So one of the alternatives is to have the regeneration using stem cells when you are to regenerate or regrow parts of your organ. So in stem cell therapy, okay, as I mentioned just now, you can opt for the stem cell therapy, means the stem cell to be used to treat certain diseases, or you can regenerate the stem cells into certain tissue or replace certain parts of your tissue using tissue engineering methods, and regenerate into various type of tissue. So for example, um, you have the donor, you have the donor. So this patient most probably is a leukemia patient. Kalau saya, if I look at the condition, this donor donating the marrow. So from the healthy donor, it could be the patient sisters, patient mother, extract marrow, and also after extract and transfer to the sick patient. Transfer to the sick patient. Okay, transfer to the sick patient. IV transplant, healthy marrow. Maybe IV transplant. Okay, healthy marrow. Okay, all right. Because other than you go for the chemotherapy, when you have done your chemotherapy, so you know that the effect of chemotherapy, when you do the chemotherapy, your cells will be gone, including the healthy cells. So to replace the healthy cells, the most easiest, because sometimes when you are sick, you cannot ask your body to regenerate cells as your as the healthy individuals. So when you have chemotherapy, it's also kill the healthy cells. So your body needs to be recovered by having a source of cells from the healthy donors. So the transplant or the transplant from the healthy donors will help the patient who undergone this chemotherapy to recover faster. And also this one, the CARDI-M therapy. So this CARDI-M therapy is to is the aim is to regenerate parts of the tissue, okay, parts of the heart tissue. Sometimes when the patient having heart disease, heart problem, and have infarction, where some parts of the hearts basically the tissue already died, infarction, due to infarction. So one of the alternative is to have it induce the regeneration or repair of the site with the stem cells. So you can basically collect the source of stem cell from bone marrow, from the bone marrow. So the collection of bone marrow is also from the patient itself, from the patient. So meaning that using in this case, his own cells. So collect from marrow, process, and get the marrow cells and transfer to the damaged heart tissue through catheter-based procedure. Dia pakai catheter and inject dekat area that already damaged, heart tissue yang damaged, and allow your body to regenerate on its own using the stem cells. So stem cell for cardiac repair, as I mentioned just now, so you can basically regenerate the heart tissue. However, you must know that heart tissue is in the category of the most difficult to be repaired, the most difficult to be defect. So yeah, but there are success stories, there are success cases. All right. So there are studies whereby they show that they can rescue the mice that having the myocardial infarction by restoring the blood flow in ischemic limbs by human pluripotent stem cells, hemangioblasts. So when we have the myocardial infarction, so your when we have myocardial infarction, meaning that our blood flow is not normal. Our blood flow is normal and we'll have restrictions going to certain parts of our body. So when we don't get the supply, the blood supply, because of the myocardial infarction, for example, in this case, in this animal, this one is mice, the myocardial infarction leads to the no blood supplies to the leg, this one. You need to imagine, you need to imagine. This one is the mouse punya tail. This one is the back leg, left and right. Tail, back leg. So if you can see in the control, this one is a control without giving. This one is all this in the control. This one is days. This one not giving any stem cell therapy, and this one with stem cell therapy. So if you see that those mice that receiving the stem cell therapy basically regenerates the affected, this one, they have no blood supply here, no blood supply, so it's gone lah. When in the long run you have no blood supply, the organ will gone, the tissue will gone. You have to amputate. Will gone, you have to amputate. All right. Yeah, I just remember one case whereby our friend's son basically undergone the brain surgery, and the blood supply does not go efficiently to the fingers, and it's end up our friend's son has to amputate the 4 fingers, okay, because of brain surgery. Okay, not of the injury, not because of injury to the fingers, but because of the brain surgery. So in this study, by giving the stem cells therapy, so this mouse basically regenerate the legs faster compared to the untreated mouse. Okay, untreated mouse. And as I mentioned just now, stem cell therapy for cartilage repair. So this is the knee, the knee. When you have injury to the knee, usually when you get older, and during your youngsters, you are too active, you're using so much of your of your foot, of your knee, then you can have a tear, wear and tear to your cartilage. Or maybe if you are too obese, so you have putting burden to your knee, and make your cartilage tear even even easier because you have more burden. Okay. So as I mentioned, when you have injury to your cartilage is not the site where it can regenerate on its own. Impossible. Okay. So one of the effort that we can do now, many clinics in Malaysia as well, you can inject the stem cells. Okay, you can inject the stem cells, so the stem cell will be there and try to regrow the cartilage on its own. Okay, on its own. And stem cell for spinal cord repair. Okay, we have 4 minutes. Stem cell for spinal cord repair, as I mentioned, you can have the stem cell from blastocyst, which is the pluripotent stem cells, differentiate into the neural stem cells, neural cells, and then you aspirate them to your spine, to induce the spinal cord repair. Okay. And there are so many clinical trials for spinal cord, especially in US especially in US. So Geron trial involving oligodendrocyte progenitor cells from human embryonic stem cells. So the advantage in US basically because they have this regulation, that's number one, and number two, they have the donor program. So it is easy for them to collect the embryonic stem cells. So yeah, you can see that the Geron stem cell asset taken over by and plan to continue with clinical trials, FDA approved first in human. Okay, so stem cell for blindness, if you can see before and after treatment, so the recovery or repair, it's much better because you are promoting the cell population in the repair area. Spinal cord injury, phase 1 and 2 in Switzerland. Okay, so stem cell treatment for stroke. Another thing, if you have stroke, there is also no turning back. There is also no turning back. If you have stroke, you have for example, the blood stroke, the bleeding in your brain. So the area is basically already injured. So to regrow, it's almost impossible to regrow on its own the brain cells. You can opt for the stem cell therapy to help regrow the injured area. Stem cell for stroke, you can have a look at this one. UK-based company Reneuron have commenced trial. Phase 1 study, 12 patients, involve proposed action through non-anti-inflammatory. So interim data by 9 patients, no adverse events. Okay, so it has a positive, promising for the future. So the last slides, I guess, challenges to stem cell or cloning research. So while applying, having pro and cons of the stem cells, so there are challenges to implement the stem cell research as well as therapy. So number one, stem cells need to be differentiated to the appropriate cell types before they can be used clinically. So as mentioned, so stem cells, to make sure that it can be used, you have to differentiate them. Okay, when you have to differentiate them, you have to grow them in the lab. As I mentioned, when you grow them in the lab, it has possibility. Of course, the cost is high because when you grow stem cells, you have to feed them with the growth factor, with the treatments, and make sure that no contamination, no genetic defects and everything. Okay, so you have to do the genetic screening, contamination screening, and so on. Recently, abnormalities in chromosome number and structure were found in 3 human ESC lines. Okay, so yeah, this is one of the challenges when you work with the cultured cells. When you work with the cultured cells, you cannot control the genetic modification in the cells. Genetic modification in the cells. So one of the things that we have to be concerned about having the stem cells from embryonic stem cells is the genetic abnormalities. So delivery to target areas. Um, okay, abnormalities, delivery to target areas. So some target areas like in brain, okay, it's very challenging to go and inject. Okay, kalau macam if it like certain even in to the heart, so you have to make sure that when you apply the cell therapy, the injection, it goes to the targeted area. So you cannot control that after you inject, you cannot control where the cells will be migrate and homing. Okay, and of course the political and religious obstruction. So as I mentioned just now, okay, it's not very easy to apply the stem cells, especially from embryonic from embryonic, due to the political and religious. So some political, religious restrictions, yeah, I don't want to touch on that, but with different religious uh, with principles and rules, there are limitations to apply the challenges, to apply the stem cells. Okay, so stem cell development or proliferation must be controlled once placed into patient. Okay, so as I mentioned just now, once you deliver, so you don't know how the cells will behave inside your body. But in certain case, like if you having osteoarthritis, you have injury to your cartilage, and you have to bear the pain, will you ever consider to think about where the cells will go? So definitely you want to opt for treatments. You don't care about how the treatment is done. Okay. And possibility of rejection of stem cell transplant as foreign tissue is very high. So this one, when it comes to the foreign tissue, meaning that you are using from the donor cells, from the banking cells, from the bank cells. So you have to make sure that it is matched genetically to avoid the rejection. And of course, as I mentioned, contamination by viruses, fungus, bacteria, and mycoplasma is very high because you conduct it in the lab environment. There are there are possibilities. And the use of mouse feeder cells to grow ESC could result in problem due to xenotransplantation. And some methods to culture the embryonic stem cells using feeder cells. Feeder cells means sometimes when you grow certain cells, you need to have other cells at the bottom as a feeder. Dia macam it gives some signals for the cells uh, to expand better. Okay, to expand better. So when you have the mouse cells, when you use mouse cells as a feeder, because mouse and human they are different species, so you can face possibility of xenotransplantation punya rejection. Okay, yeah, I thank you very much. Any questions? Any question from you? It's, yeah, sorry, I actually exceeded 4 minutes. Uh, Okay, so all right. If you have no question, I would like to touch on the presentation. So I have posted in the e-learning. There are few topics given to you. So you are requested to come up with a presentation. Okay, you are presentation is a 10-minute presentation, maximum 10-minute presentation. You can select any topics that I have listed. Okay, so it is individual presentation. Okay, so please prepare it in a video format, and please submit the presentation via e-learning. So we will not have we will not have a separate presentation slots for that. So you just need to submit via e-learning. Okay, so it is 10-minute presentation. So make sure that you put your name, introduce yourself, okay, and also um, uh, please prepare it with yeah with interactive presentation, okay, with interactive video, because the marks will be given based on the presentation and the contents that you included in your presentation. So you are given 2 weeks, okay, please submit in 2 weeks. I already put the deadline in the e-learning. So please refer to the instructions. And if you have question, your class rep can email me or can reach me via my phone number. So if you have no more questions, thank you very much for your presence today. So I hope that you can complete your assignments within 2 weeks. Um, okay. Yes, Yin? We still have no class on Friday, because I think I was given two slots. Another slot is for presentation. Because if we would like to have presentation on the second slot, I should give you the topic much earlier. I should give you the topic much earlier. It's unfair for you to prepare in 2 days. I know that you are busy. Okay, you are busy, pile up with all the assignments. So I'm not that cruel to ask you for the to ask you for the presentation to be submitted in 2 days. So I give you 2 weeks. So please submit your presentation in 2 weeks. Okay. All right. Thank you very much. Have a nice evening. Thank you. Bye.